was reading about famous biochemists looking for topics for this channel, and I came across the work of Eric Kandel from Columbia University. He won the 2000 Nobel Prize in Physiology or Medicine due to his work on the physiological basis for memory storage. I ended up reading a 23-page review written in 2014 by Eric Kandel and a few other neuroscientists titled The Molecular and Systems Biology of Memory. I'll go through what I learned about the biology and chemistry of learning. This covers some of the 250 articles cited in this review. Before reading this review, I understood how computer memory worked better than I understood my own memory, which is sad to think of as a biochemist. Ones and zeros are used to store information, and you need a processor to encode the information and organize it. You also need a way to retrieve the information. Does memory in humans work the same way? In a way, yes. I learned that the synapse strength is the secret to encoding memory. But I went through the tome of a paper to figure out how the connections are made and maintained. The idea that memories were stored and the variation of neural connection strength was first suggested by Santiago Cajal, who won the same Nobel Prize as Eric Kandel did in 1906. If you need a refresher on how neuron firing works, check out the videos below on synaptic transmission and action potential. Human memory code is more like the alphabet than binary because the various flavors of neurotransmitters and chemistry are like letters, and the various strengths of a type of firing are like lower and uppercase. Memory can be categorized into implicit or explicit memory. Implicit memory involves perceptual and motor skills. Implicit memory doesn't involve any conscious awareness. Explicit memory includes places, facts, objects, people, and does involve conscious awareness. There's so much to unpack, so I'll start with implicit memory in this episode, and then I will explain explicit memory in the next episode that is perhaps more relevant to our own brains. I will put the link in the description for this video once that video is out. The review focuses on Aplysia slugs. These sea slugs are so simple biologically and neurologically, and they have huge brain cells measuring up to a millimeter. So we have a thorough understanding of their molecular biology. Later, similarities were described in mammals. Short-term memory creation or facilitation in Aplysia, such as measured gill withdrawal reflex, results from a change in transmission of neurotransmitters from the presynaptic terminal to the motor neuron. The memory can resemble habituation with a decreased flow and synaptic weakening, or sensitization, an increased flow and synaptic strengthening. Sensitization occurs with activation of modulatory neurons, which release serotonin into sensory neurons. This elevates cyclic AMP and sensory neuron. We'll call it CAMP. And this directs the presynaptic neuron to release more of the neurotransmitter glutamate into the synaptic cleft, making it stronger. Mutations in the CAMP pathway worsen short-term memory in Drosophila, which can learn fear, in agreement with Aplysia results. Glutamatergic synaptic connections, meaning connections that are activated with glutamate. These connections in neurons were put on 2D slides. This is called dissociated cell culture. And these connections showed a short-term strengthening when serotonin was added. Repeated application of serotonin produced synaptic strength that lasted more than a week. Repeated release of serotonin that happens with long-term sensitization gives rise to protein synthesis in Aplysia. CAMP-dependent protein kinase, or PKA, recruits mitogen-activated protein kinase, or MAPK. This is a kinase that's involved in the signaling to induce transcription. I studied inhibition of a kinase in a MAPK pathway called P38-alpha in my first semester in grad school, which resulted in a publication. PKA recruits the transcription factor CAMP response element binding protein, or CREB1. By blocking binding to its DNA response element, long-term facilitation was prevented. Immediate response genes express because of CREB1 that keeps a PKA involved in short-term facilitation. CREB1 also induces transcription factor CCAAT enhancer binding protein, essential for long-term facilitation. This transcription factor expresses genes that produce growth of new synaptic connections. CREB2 is a memory suppressor gene, preventing long-term facilitation, which opposes the function of CREB1. CREB-mediated response pathways can be modulated by other phosphatases and kinases. So this integrates signaling from other pathways. In addition, there is a microRNA strategy for the inhibition of CREB1 and CREB2. Click the link below if you want to learn more about microRNAs. Epigenetic marking in response to other stimuli regulates expression at loci that allow for long-term synaptic chain. CREB proteins also regulate expression through the acetylation and deacetylation of histones, which affect the nucleosome structure. Habituated aplesia retract synaptic terminals and make fewer connections with motor neurons or interneurons. The opposite happens with sensitized animals. Details have been worked out. Growth starts with the presynaptic neurons. PKA and presynaptic neurons release glutamate at threefold the normal level, activating metabotropic glutamate receptors, mGlu-R5, in the postsynaptic neuron, which increase inositol triphosphate, or IP3, which releases calcium. Calcium will allow for the insert of new copies of amino-methylpropionic acid, glutamate receptor, into the postsynaptic terminal, which is the first phase leading to synaptic growth. Holy moly, I'm so sorry. I actually thought that was interesting when I read it, but now I sound like my high school biology teacher. Let's move on. Now this part is really cool. Long-term facilitation has been shown to be synaptic specific. Synapse specific, meaning that 1 and sometimes 1,000 synapses in a neuron is strengthened. Now, how does that happen? When protein synthesis is required. In aplesia, serotonin was applied to one of 2 branches of a bifurcated sensory neuron that innervate 2 motor cells. The change persisted for 24 hours in that one neuron that was exposed to serotonin. This implies that signals went to the nucleus and then back to that specific synapse because protein synthesis is required for long-term facilitation. Before I conclude, let's focus for a second on implicit memory in mammals. The brain areas involved here are the cerebellum, striatum, and amygdala. The conditioned fear response in mammals involves the amygdala that receives auditory input from the thalamus and process info from the neocortex and gives it output to the hypothalamus that directs autonomic fear responses. Like long-term facilitation in aplesia, neurons in the amygdala can undergo increases in synaptic strength in response to repeated stimulation. A conditioned fear stimulus, like a sound that was previously paired with an electric shock, goes to the amygdala. Synaptic alteration and maintenance of memory for learned fear also needs PKA, like aplesia, as well as MAPKs and activation of CREB. Altered synapses also require the recruitment of AMPA receptors. Altered synapses also bring AMPA receptors to synapses of amygdala neurons. Learned safety, similar to habituation in aplesia, involves lowering of auditory input to amygdala and opposes learned fear. Research in mice is showing that any neuron can be activated, but the resting state is important. Introducing excess or constitutively expressed CREB, which means continually expressing the transcription factor, into certain amygdala neurons in a fear conditioning study resulted in those specific neurons encoding memory. This shows the flexibility of neurons activated, and it shows that the resting state is important. Scientists were able to identify and activate cells that were responsible for the fear conditioning response. They engineered rodents to express channel rhodopsin, CHR2, in neurons activated in a fear conditioning study. Light pulses on their brain activated those particular neurons because channel rhodopsin is a light-activated ion channel, and fear was observed. That brings this episode on implicit memory to a close. If you remember anything, just know that synapse strength is what encodes memory. We'll find this to be the case in my next episode. on explicit or conscious memory in mammals, but we'll dive into the function of brain regions that encode and retrieve information. Stay tuned.